This PowerPoint presentation talks about development of the nervous system from the beginning to the end of prenatal development, and also talks about postnatal development, that means after birth, and also talks about some developmental disabilities. On this slide you can see the neural plate, which is shown in yellow, which develops along the posterior surface of the embryo, and this curls up to form the neural folds, and the neural folds then touch each other to make a neural tube, which is in the shape of an O. So the neural plate rolls up to form the neural tube. Here's another view showing the neural plate, which is now in blue, curling up to form the neural tube. You can also see in the bottom of this slide the red neural crest cells, which pinch off from the sides of the neural tube as it forms and migrate throughout the body to form the autonomic nervous system and also to form the dorsal root ganglia. So the peripheral part of the sensory nervous system is formed from these neural crest cells. The structure that's inside the red circle on this slide is an example of the neural tube, and it's shown in a scanning electron micrograph, so this is a picture of the real thing. The layer of tissue that's just above that forms the skin of the back of the embryo. After the neural tube forms, it subdivides into regions called forebrain, midbrain, and hindbrain. And in this slide you can see all the different parts of the tube as they develop and differentiate, shown in different colors. And you can see that the tube starts to get subdivisions and bulges in it. The part at the very top, called the telencephalon, which is in orange, is what's going to form the forebrain. The midbrain is shown in red, and then the hindbrain is shown in green, and then the spinal cord is inferior to that. So all of these structures are formed along the posterior aspect of the embryo, covered up by the skin of the back and the vertebrae. And in the bottom of the slide you can see what these structures ultimately end up in, a fully developed brain. This slide lists for you the structures that form from the neural tube and from the neural crest. So the neural tube forms the brain and spinal cord. The neural crest, the cells that migrate throughout the body, form the dorsal root ganglia and the autonomic nervous system. This slide shows the parts of the brain that form from the neural tube in more detail. So prosencephalon is what's really going to give rise to the forebrain structures, the cerebral cortex, the basal ganglia, the thalamus and related structures. The mesencephalon is the midbrain, and that gives rise to the midbrain. And then the rhomencephalon means the hindbrain, which gives rise to the pons and cerebellum as well as the medulla oblongata. This slide shows the development of the brain at various stages of development from the 2nd month all the way through the 7th month prenatally. And what you can see mainly in here is how the telencephalon, or forebrain, undergoes the largest amount of growth and also of folding and changing. Here we have a slide that shows the development of the spinal cord. So dorsal is towards the top, and ventral or anterior towards the bottom of each of the 3 pictures. And what is described here as the alar plate, the blue structures, are going to give rise to sensory neurons and sensory structures. And the pinkish purple stuff, the basal plate, on the ventral or anterior part of the cord is going to give rise to the motor neurons. So the motor neurons are found on the ventral side, and the sensory neurons are found on the dorsal side. This slide shows ventral neurons coming from the ventral or anterior part of the gray matter of the spinal cord, and sensory neurons from the dorsal root ganglion coming into the dorsal part or posterior part of the spinal cord. This slide also shows some interneurons, which are shown in black, which develop a little bit later and which connect sensory and motor neurons to form a reflex. During development, there are 4 major processes that neurons undergo as they're going through the process of growth and development. Those are listed here. Mitosis, which is cell division, that's creating more neurons. migration, which is cell movement, in which neurons move to the correct locations in the nervous system, differentiation, which is another term for specialization. That means taking on a specific identity as an interneuron or a motor neuron or some other thing. And finally, myelination, which is the process of laying down myelin along the axons. All four of these processes take place prenatally, in other words, before birth, and myelination is the only one that extends postnatally, so after birth. That means that mitosis, migration, and differentiation occur only prenatally. Mitosis is the process of cell division, taking one cell and making 4 smaller cells from it. This is the process by which new neurons and glial cells are created. And for neurons, this occurs only before birth. And any drugs that a pregnant woman might take, for example, chemotherapy drugs or other so-called recreational drugs, can interfere with this process and cause defects in the fetus's brain. Neurons have to move or migrate to the correct locations within the nervous system, and they're guided by chemical cues. Some of these cues say, come here, others say, go away from there. And so with both attractive and repulsive sorts of stimuli or chemical cues, the neurons are guided to the correct locations within the nervous system. If they fail to migrate to the right places, then there can be defects. Some of these include Hirschsprung's disease, which is described in the next slide, and this is also possibly connected with seizure disorder, in which neurons are found in the incorrect locations within the brain. Neurons need to differentiate or specialize into a special function. This is typically based on their location. So if migration does not occur correctly, then differentiation can't occur correctly either. People with Down syndrome have brains that contain a lot of undifferentiated neurons that have not specialized into one function or another, which is one of the reasons that they have cognitive disabilities. The process of myelination begins prenatally and does not fully complete until about age 20 or 21 once we're born. It is the basis of postnatal brain growth. So when you take your child to the doctor and the doctor measures your baby's head, what they're really looking for is to make sure that the brain is growing, and most of that growth is based on the growth of glial cells, which are in the brain, oligodendroglial cells, and which are responsible for most of the growth of the brain postnatally. This slide is just here to remind you of the function of the myelin sheath. It increases transmission speed of the action potential by insulating the axon so that the action potential is able to move from one node of Ranvier to the next in a phenomenon called saltatory conduction, which means that if a child's brain does not myelinate correctly, action potentials will not be transmitted rapidly within the nervous system. This slide describes the state of the nervous system at birth. So we have many neurons. In fact, more neurons are found in the newborn baby's brain than in the brain of an adult. But there are very few synapses or connections between these neurons. Sensory neurons are typically pretty well myelinated. Although babies have trouble localizing sensation exactly, they are aware of pain and temperature and pressure and other types of sensations. They're also good at smelling and tasting. In the motor system, nerves that control muscles for sucking and swallowing are well myelinated, but most other nerves in the motor system are not well developed, which is one of the reasons that newborn babies can't walk and talk and move in a coordinated way. That's stuff that they have to learn, and as they learn that, the myelin will grow along their axons. During the first year of a child's life, there's a tremendous amount of growth of the brain. It should double in size, all of which, or most of which, is due to proliferation of the oligos and the thickening of the myelin sheath that those oligos are responsible for. The brain uses 60% of the energy that the child consumes, which is why they get you up all night long in order to nurse. There are two major developmental processes that occur postnatally, in other words, after the baby's born. These are myelination and synaptogenesis. Synaptogenesis means the growth or birth of new synapses between existing neurons. This slide describes the time frame for myelination to be completed within the central and peripheral nervous systems. As you can see here, the tracts in the spinal cord are mainly myelinated by about a year of age. Motor and somatosensory cortex take a little bit longer, should be complete by about 16 months of development, and the rest of the CNS is mostly myelinated by age 2. However, some areas take a lot longer to finish myelinating, and those include the prefrontal cortex and the corpus callosum. We haven't studied what the function of the prefrontal cortex is, but I can tell you that one of its major functions is to allow you to solve problems, to think through the consequences of your actions, and to inhibit impulses. And that is not completely myelinated in most people until about age 20, a little bit later in males than in females. So think about some teenagers you know, and think about the fact that their prefrontal cortex is not fully developed yet, and that might explain a lot. Synaptogenesis occurs from birth through death, so it is an essential part of postnatal development. A tremendous amount of this occurs during infancy and childhood. And synaptogenesis is the molecular basis, the biological basis of learning. This creates new connections and pathways between existing neurons and requires appropriate sensory stimulation. There are critical periods that appear to exist for vision, hearing, language, and proprioception, and those neurons that don't make connections during this time are pruned away or removed, which is why, although we are born with many neurons, we do lose some as our brain grows and develops, and that's a perfectly normal process. This slide and the next series of slides show some developmental disorders. This horrible picture is a picture of a baby with anencephaly. This means without a brain. So in this case, the neural tube failed to zip up or close in its superior direction, and so it's open at the base of the neck and the skull. And essentially the only part of the brain that this child has is a brain stem. So it's usually described as without a brain, or it's said that the brain does not form. The baby is either born dead or dies shortly after birth. In spina bifida, the neural tube fails to close in an inferior direction, and that leaves the spinal cord open and unprotected by the vertebral arches, and sometimes even by the skin on the back, as you can see in this picture. The bottom right picture shows different forms of spina bifida. On the left, you can see that there's just a vertebra missing, but the spinal cord itself is in place. The middle picture shows the spinal cord in place, but a sac of meninges sticking out of the back. And then the picture on the right shows that the spinal cord and spinal nerves, the cauda equina, are actually protruding through the back in that sac of meninges. So if this is not corrected surgically, the child will die of infection. If it is corrected surgically, what typically happens is that the child has paralysis from whatever the site of the lesion is inferiorly. And functionally that looks very much like spinal cord injury, although it's not caused by trauma, it's caused by a birth defect. Here's another picture showing spina bifida. This one is a little bit more inferior than on the previous slide, and so this neural tube closed up further before it remained open, but you can see that sac of meninges, the dura mater, and arachnoid poking out from the baby's back, and you can see that the spinal cord and spinal fluid are present in that sac. Hydrocephalus, or water on the brain, is caused by blockage of the flow of cerebrospinal fluid. This can accompany spina bifida, although it doesn't always. And what will happen is that the ventricles of the brain enlarge dramatically. You can see that in the picture on the bottom right, which shows a normal and hydrocephalic brain. And because the baby's bones in a newborn child, the baby's skull bones have not fused together, the skull bones will enlarge as well as the brain. And so in the bottom left, you can see the picture of a little tyke there who has a greatly enlarged skull. And then if you look at the two pictures of the skulls, you can see that they're greatly distended as well. This always causes major cognitive, motor, and sensory deficits if it's not corrected. Arnold-Chiari malformation is a condition in which the cerebellum and brainstem are herniated down into the cervical spine, and you can see that on the pictures. Because your brain does not belong in your neck, what will happen is that the medulla oblongata and cerebellum are compressed within the vertebral column, and this can cause cerebellar symptoms such as lack of coordination or ataxia, as well as medullary symptoms, which can include cessation of breathing, because the respiratory nuclei are located in the medulla. This is usually corrected with surgery, in which the brain is pushed back up into the skull and then some kind of bone graft is put into the neck to prevent the herniation of the brainstem and cerebellum back down into the neck. Down syndrome is a genetic condition in which there is a third 21st chromosome. Please do not worry about the upper right diagram. It's just there to show you that we do understand what happens at a genetic level in Down syndrome. People with Down syndrome have a number of physical conditions, and neurologically they tend to have a large number of undifferentiated neurons and fewer synapses. And synaptogenesis in people with Down syndrome is much slower than in other people. So people with Down syndrome can learn, but it takes them longer to learn the same things than it would a person without Down syndrome. Fetal alcohol syndrome, or FAS, is a completely preventable developmental disorder in which excess alcohol consumption by a pregnant woman disrupts many aspects of brain development in the developing child. The brain tends to be small and malformed. You can see that if you take a look at the brain photos on the right side of the screen. And there are also a number of other syndromes that you can see if you look at the photos. A small jaw, thin upper lip, low nasal bridge, and so on. Don't worry about those things, but what you want to know is that alcohol disrupts all different kinds of aspects of brain development. Cerebral palsy, or CP, is defined as a motor disorder that occurs before, during, and after birth. So it can be caused by lack of oxygen during delivery, it can be caused by infection, it can be caused by a small stroke that a baby has while being born or shortly after birth. All of these things are lumped together as cerebral palsy, and it is defined based on its motor symptoms. However, other things such as sensory, cognitive, visual, and hearing deficits often accompany the motor dysfunction. This slide lists for you the 4 types of cerebral palsy. They include spastic, ataxic, apoptoid, and mixed. Spastic cerebral palsy is the most common and is caused by damage to upper motor neurons. And as you know, damage to upper motor neurons causes spastic or hypertonic paralysis. It's very common for this to affect one side of the body, which is called hemiplegia. It can sometimes affect both lower extremities. That's called diplegia. So spastic CP is the most common type. It is also possible to have damage strictly to the cerebellum. That would cause ataxic cerebral palsy, or damage to the basal ganglia, which causes apoptoid cerebral palsy. This is characterized by uncontrolled writhing movement that the patient cannot inhibit. If more than one type of CP is found in the same person, so for example, if somebody had both spastic and ataxic cerebral palsy, that would officially be known as mixed. Notice that all 4 of these are based on the motor disorder, regardless of what other kinds of visual or cognitive or other types of deficits might occur in the patient. Autism, or pervasive developmental disorder, is a very complex condition with a wide range of social interaction disorders. It is genetic. There are over 20 genes that have been identified so far, and more are being identified all the time. I will mention at this point that there is absolutely no evidence that it has anything to do with vaccines. That's been disproven. Some of the characteristics are listed on the right side of the screen. Basically, autism is considered a disorder of social interaction. And other than the genes, we don't know exactly what causes it. It is possible that there is some sort of environmental trigger, but exactly what that is has not been identified. This slide lists some of the differences between the brain of a person with autism versus the typical brain. People with autism have a larger brain than others with more neurons. However, their cerebellum and brainstem are significantly smaller. They have fewer neurons that produce serotonin, and in fact, serotonin reuptake inhibitors, or SSRIs, can be useful in some people with autism in treating the signs and symptoms. And there is also decreased activity in parts of the cortex that process auditory signals and facial recognition. Our last slide in this presentation describes epilepsy, or seizure disorder. This is a condition in which there are multiple reoccurring seizures. It's possible to have one seizure for example, from a brain tumor or a CVA or something, but that does not constitute epilepsy. Epilepsy is where there are recurring seizures. And these can be caused by neurons that are found in the wrong place within the brain as a result of improper movement of neurons. It can also result from genes that cause increased depolarization of neurons or a deficit in the inhibition of action potentials, so it can be genetic. It can also sometimes be caused by an infection that damages part of the brain, or a traumatic brain injury or a stroke that damages part of the brain. Epilepsy in general is treated with inhibitory medications, things that increase the activity of inhibitory neurotransmitters. As a result, many of those treatments will cause inhibition of typical cortical function, and therefore fatigue is one very common side effect.